Cellular respiration is the breakdown of glucose into carbon dioxide, water, and energy in the form of ATP.
In glycolysis, there are two phases, energy investment and energy produced. In the energy investment phase, 2 ATPs are changed into 2 ADPs and phosphate groups. Later on in the cycle, 1 NAD plus, 2 hydrogen ions, and 2 phosphates are changed into 2 NADHs. At the end of the cycle, there are a total of 4 ATPs, with a net gain of 2. Two pyruvates are also produced from glycolysis. Aerobic respiration is a process that requires oxygen to perform inside the cell. Anaerobic oxidation is a process that we will see later on in the video, which does not require oxygen and happens right after glycolysis.
After glycolysis, the two pyruvates go into the process called pyruvate oxidation. They're transferred from the cytosol to the mitochondria via the transport protein. In this process, CO2 is given off from the pyruvate. NAD plus is converted to NADH and H plus. Coenzyme A attaches to the acetyl group, and acetyl-CoA is formed as a final product of pyruvate oxidation.
Anaerobic fermentation, or alcoholic fermentation, is a process that occurs after glycolysis. During the process, 2 pyruvates are broken down into 2 ethanols, and a CO2 is released. At the end of the cycle, 2 ATPs are produced as a result of the 2 pyruvates being broken down.
Lactic acid fermentation is another form of anaerobic fermentation. This occurs after glycolysis. Pyruvate is broken down into lactate. Since there are 2 pyruvates, there should be 2 lactates at the end. NADH and a proton is also turned into NAD+ in this process.
After pyruvate oxidation, acetyl-CoA starts the cycle of the Krebs cycle. After the whole thing is completed, 2 CO2 are produced, 3 NADHs, 1 FADH2, and 1 ATP. In oxidative phosphorylation, there is an electron transport chain. NADH and FADH2 are oxidized to provide electrons for the chain. FADH2 is converted into FAD. The electrons are finally passed to the terminal acceptor O2 at the end of the chain, forming water. The O2 combines with the 2 hydrogen ions to form this water. During chemiosmosis, ATP synthesis is powered by the flow of the proton gradient in the electron transport chain across the membrane. The ATP synthase uses a proton gradient power to phosphorylate ADP to form ATP. Substrate-level phosphorylation occurs in the cytoplasm of glycolysis, pyruvate oxidation, and the Krebs cycle.